Welcome to Ramya Teacher's Classroom. Today we're totally unraveling a core mystery of plant biology. We hear it all the time, right? Plants breathe in carbon dioxide and breathe out oxygen. But, well, in the world of science, we don't just take things at face value. We've got to actually prove them. So, in this explainer, we're going to step into the shoes of early biologists to figure out exactly how we know, like beyond a shadow of a doubt, that carbon dioxide is an absolute, non-negotiable ingredient for a plant's survival. Here is our lesson plan for today's explainer. We'll start off with an intro to plant nutrition, check out the ingredients for photosynthesis, dive into the missing ingredient mystery, look at Moll's half-leaf experiment, and finally wrap up with the carbon dioxide connection. Let's get to it. Section 1, Intro to Plant Nutrition. How do plants actually eat? Okay, let's dive into this. Every living organism needs energy, but green plants, they have this pretty amazing trick up their sleeve called autotrophic nutrition. Basically, auto means self, and troph means nourishment. So unlike us, wandering over to the fridge looking for our next meal, plants are kind of like their own self-nourishing personal chefs. They actually synthesize their own complex food right on the spot, just using simple inorganic raw materials. The soil gives them water and minerals, and the atmosphere provides, well, actually, that's exactly what we're going to prove today. Moving on to Section 2, Ingredients for Photosynthesis. What exactly gets put together? So this whole food-making process is called photosynthesis, which literally translates to putting together with light. But what exactly is being put together here? Well, scientists have experimentally proven that there are 4 non-negotiable conditions for photosynthesis to happen. First up, you need sunlight. That's what provides the actual energy. Second, you gotta have chlorophyll, which is the green pigment that acts sort of like a biological solar panel to trap that light. Third, you need water, absorbed by the roots. And fourth, you need carbon dioxide. If you take away even just one of these 4 pillars, the entire food production factory literally shuts down. Now, water comes from the ground, right? But carbon dioxide is a gas in the air, so how does it actually get inside the leaf? Well, it happens through stomata. These are tiny, microscopic pores located mostly on the surface of the leaf. And it's pretty cool. Each pore is guarded by 2 kidney-shaped cells that swell and shrink to literally open and close the hole. When they open, gaseous exchange happens. The plant literally inhales carbon dioxide from the surrounding atmosphere right through these tiny doorways. Which brings us to Section 3, the missing ingredient mystery, testing CO2's exact role. So what does this all mean? It's one thing to look at a diagram, but it is an entirely different thing to prove the plant is actually using that gas. So here's the puzzle we're going to solve together. How do we actually prove that plants absolutely need carbon dioxide to survive? I mean, if CO2 is everywhere in the air around us, how on earth can we test a plant's ability to survive without it? We can't exactly just stick a plant in a total vacuum. It would die from a lack of oxygen and pressure. So we need a clever way to selectively remove just the carbon dioxide from the plant's environment. Enter our secret weapon for this experiment, potassium hydroxide, or KOH. This chemical is absolutely fascinating because it acts almost like a sponge, but specifically for carbon dioxide. If you put some potassium hydroxide in an enclosed space, it will aggressively absorb all the carbon dioxide present in that trapped air. This gives us the perfect tool to essentially steal the CO2 away and create a completely carbon dioxide-free zone for our test. But before we actually run the experiment, we have to prep our plant using a very specific sequence. First things first, we must de-starch a potted plant by leaving it in complete darkness for about 3 days. Now, you might be wondering, why do that? Well, plants store their leftover food as starch. If we don't completely empty the plant's pantry first, our test is going to be ruined by old food. After 3 days in the dark, the leaves are totally empty of starch. Then, we expose the plant to our experimental conditions. Finally, we treat the leaf with an iodine solution. The logic here is simple. If the iodine turns a deep blue-black color, starch is present, which means the plant successfully performed photosynthesis and made new food. If it doesn't turn blue-black, no food was made. All right, Section 4, Moll's half-leaf experiment. Let's look at this incredibly elegant setup. Now, what's really interesting about this slide is how simple but brilliant it is. Armed with our potassium hydroxide and iodine, we're ready for one of the most elegant physical setups in biology. The sheer simplicity here is just great. All you need is a wide-mouthed glass bottle and a split rubber cork. By splitting that cork in half, early scientists realized they could insert a single, living leaf halfway into the bottle without actually crushing its stem. This perfectly isolates one half of the leaf inside a controlled, tiny microenvironment, while the other half just stays outside in the normal, natural world. It's literally the ultimate side-by-side comparison, all happening on the exact same leaf. Okay, let's meticulously walk through the steps of Moll's method. Step 1. We place some of that potassium hydroxide right inside the wide mouth bottle. Step 2. We carefully insert just the tip half of our pre-destarched leaf inside the bottle, sealing it tight with that split cork. Step 3. The base half of the leaf is left sticking out, completely exposed to the regular atmospheric air. And step 4. We place this entire delicate setup in bright sunlight for about 3 days. So think about what's happening here. The whole leaf is getting sunlight, the whole leaf has water coming up from the roots, the whole leaf has chlorophyll, but thanks to the potassium hydroxide acting as a sponge inside the bottle, only the tip half of the leaf is being completely deprived of carbon dioxide. So after 3 days, we remove the leaf, we dip it in boiling water and ethanol to strip away all that green color, and then we apply our iodine drops. And wow, the results are dramatic and completely undeniable. The base half of the leaf, the part that was outside the bottle, breathing normal CO2-rich air, immediately turns this deep, dark blue-black color. But the tip half of the leaf, the part that was trapped inside the bottle with the potassium hydroxide, it doesn't change color at all. No way. It just remains totally pale. Which takes us to section 5, the carbon dioxide connection. Let's interpret what this actually means. We've seen the color change, right? Now let's really cement the science here. So the crucial point is this. The only difference between the top half and the bottom half of that single leaf was the presence of carbon dioxide. Because the iodine only turned blue-black on the outside half, we know for a fact that starch was only synthesized where carbon dioxide was present. The tip, inside the bottle, had light, it had water, it had chlorophyll, but without CO2, it was basically paralyzed. It couldn't make a single molecule of food. Moll's half-leaf experiment doesn't just suggest that plants use carbon dioxide, it provides definitive, visual proof that CO2 is an absolute, non-negotiable requirement for photosynthesis. We've just proven on a micro level how completely dependent plants are on the carbon dioxide in the air, but let's zoom out to the macro level for a second. Through this exact process, plants are constantly purifying our atmosphere. They are consuming massive amounts of carbon dioxide and releasing the oxygen that you and I are breathing right this very second. It's this perfect biological trade. But, it leaves us with a pretty profound question to ponder. As human activity drastically alters the levels of carbon dioxide in the atmosphere and deforestation reduces the number of these autotrophic kitchens available to process it, what happens to this delicate, ancient balance that keeps our entire biological world fed and breathing? Definitely something to think about until our next explainer.